We're going to preview another award that's going to be happening tonight, and to do that, I'd like the Chief Scientific Officer of the UMDF, Phil Yeski, to join us.
Yeah, so I'm here to talk a little bit about the foundation. I always share with my colleagues, LHON is a mitochondrial disease. Mitochondrial disease is UMDF. That means LHON is UMDF. So we want you to feel a part of this community. You are a part of this community. And we're always looking for ways to help build those bridges and make those ties even closer. Our grant program, as you may have heard, refocused this year, and we ran a cycle where we asked for very specific kinds of proposals around biomarker development. And biomarkers are representative of the disease, so ways we can measure the disease, sometimes directly, sometimes indirectly. And as it turns out, through a very rigorous peer review process, we ended up selecting an LHON-focused award. So now we would like to bring the recipient of that award, Rustam Karanjayed, up to the podium and make a small presentation. Woo! Yeah, maybe you should say a few words.
Dr. Karanja's a little bit later. He's got some slides, so he's going to tell you a little bit more about the actual research he's going to do. So we'll do that later. But
yeah,
if people can't read what the award says, this is an award that was co-funded through, some of you know that we do an LHON project fund, and we partnered with the UMDF. It was largely, it was a full cycle through UMDF, but again, it was LHON related, so we helped. So the amount on the check is
$100,000.
Holy shit.
One of the first grants that the LHON project gave was for work done by Dr. Sadoun's team, as well as Dr. Karanja, for an LHON mood study. And if you were at last year's conference, you heard Dr. Karanja talk about the results of the study. And so that video is on that list of videos from prior conferences. So we funded that project back in 2014. Dr. Karanja presented the results at last year's conference, and then since then a couple of papers have been published. So it's exciting when our group raises money, gives it to researchers, things get published, and there's more work actually in development, more papers yet to be written. You saw earlier the notion of when UMDF does its grant cycle, and they study, they take all kinds of folks to receive grants, and they do a comprehensive review process. And when they do select a project that's LHON related, they do reach into the LHON project and ask if we'd like to co-fund. And back in 2015, there was a grant given, and the person who received it, I just wanted you to hear a little bit from Dr. Atif Tawhid about what he did with the award he won back in 2015.
Thank you so much, Lisa, for this wonderful opportunity. My name is Atif Tawhid, and I am a postdoctoral fellow with Dr. Doug Wallace at the Children's Hospital of Philadelphia. My project focuses on evaluating a unique gene therapy approach for LHON. Leber's hereditary optic neuropathy has features which involve sudden onset blindness in 20s and 30s, resulting in central scotoma or vision loss, and it is caused by mitochondrial DNA mutations. The panel on the top right-hand corner shows the eye of the affected individual. Below that is just to show the complexity of the transmission of the disease and that it is maternally inherited with a male bias. On the left-hand panel, I'm not going to go into the sequences of mitochondrial DNA sequence, but I would like to mention that my project is focused on a unique cell hybrid model that was developed in the Wallace lab that has a mutation in ND6 gene. ND6 gene that is encoded by the mitochondrial DNA is known to cause 15% of the LHON cases. So I won't be going into a lot of details with the results and the backgrounds, but I thought it would be great to share two key pieces of data that I have gotten so far using the gene therapy vectors that we have developed in the Wallace lab. This, as a summary, we find cells with mitochondrial dysfunction showing phenotypes closer to normal cells after the gene therapy rescue. In the left-hand panel, it's the mitochondrial content, and then there are four bars in that graph. The extreme left, the black-colored graph, is the control cells, and this is the mitochondrial content, how much mitochondria is present in the cell. The second graph, which is red in color, is the mitochondrial mutant cells, and we can see that it has more mitochondria, and we would expect that because the cells are lacking ATP, and the cells tell the mitochondria that you need to make more ATP, but because it has a mutation, it is unable to. So as a compensation mechanism, it undergoes biogenesis and it creates more mitochondria. The next two bars that we see are the results of mitochondrial content after the rescue from human, after the rescue using the human ND6 and the mouse ND6 genes. And it is evident that both those constructs are able to bring the mitochondrial content closer to normal, and that's what we want. The right-hand panel is the rate of lactate production. Again, the black bar is the normal cells, and the red, which is the mutant cells, has almost double the rate of lactate production. When we treat it with the gene therapy construct, at least one of them shows that it brings it back towards the normal levels. The last one is still undergoing testing, and we'll have more data. So currently, we are investigating more biochemical and physiological assays. The ultimate goal is, once we have one or two vectors that is working perfectly, we will transition them into an AAV vector system, which will then be injected and evaluated in an LHON mouse model that Dr. Wallace's lab has developed. So that's all that I have to share. I would like to acknowledge my mentor, Dr. Doug Wallace. He's a great mentor and one of the brilliant scientists, and it is my absolute honor to work with him. My esteemed CMEM members, a few of them who are sitting in the back, thank you so much. And of course, our collaborators, and all this wonderful project and collaborations are possible because of the UMDF and LHON project that fund my project, and of course, to such a wonderful and vibrant community. Thank you so much. You energize our project.
Thank you. And as you heard earlier, this year, the UMDF again selected an LHON-related project, and again, the work that was done by folks in this community to raise funding has helped co-fund that project, and Dr. Kranja is going to just give you a very brief overview of what he's going to be working on.
How many of you have been to see a neuro-ophthalmologist? Can you put up your hands? And how many of you have done a visual field? And how many of you hated doing the visual field? Okay. So that basically is why we're doing what we're doing. So essentially, what we're looking at is we're trying to figure out a way to look at the data that we're getting from individual patients to try and understand what's going on in the disease in a way that allows us to be able to do interventions through clinical trials or through other methods to see how the progression of the disease is going over time in a reliably reproducible way. And eye diseases are really interesting and fascinating as a group, but it's a real challenge as an ophthalmologist to figure out what's going on in the eye. Most of the tests that we do with patients are subjective. They all involve you inputting and giving us an answer to a question. And when we do that, we end up with a large range. Your vision is going to fluctuate from day to day. A normal patient's vision is going to fluctuate from day to day. And if you're really tired and you get stuck inside that machine trying to do the visual field, you're going to do a lot worse job than if you were bright, happy, and well awake. So because of that, when we get into situations where we're looking at things like this, where we've got very dense central scotomas, we've got issues where the machine now is no longer within its receptive range, it's reached the bottom of its receptive range, it's going to have difficulties trying to pick up changes, it's going to try and have difficulties trying to pick up improvement. When we try and look at measures that don't rely on input from patients, we really have two different types of measures that we can look at. The first type of measure essentially is looking at the anatomy. We can look and see how the anatomy is changing over time. In that situation, we do something called an OCT-RNFL. And when there's also another variant of it, it's called a ganglion cell complex. And both of them are basically, you sit in front of a machine, it scans the back of your eye, and it takes a cross-sectional image of the back of your eye that then is converted into a measurement of the thickness of the retinal nerve fiber layer. Now, there's a challenge in Leber's in that there's swelling going on at the same time as the atrophy. So it's kind of hard to track an acute patient when they are going through the change from being a carrier to affected using OCT as an outcome measure. The other side of this is electrophysiology. And some of you who visited us at UCLA have been through these tests that we're talking about, and you've seen what we do. And essentially, what it involves is looking at a stimulus, either a light that flashes or a pattern that goes back and forth. And both of these have their advantages and have their disadvantages. But essentially, they're recording from the retinal ganglion cells. They're recording from the ganglion cells, and that is the component of the electrical activity that we're interested in. So the first one involves looking at this pattern that goes back and forth. But you can imagine that trying to look at this little X here, the red cross, is not the easiest thing to focus on. It's kind of like doing a visual field, but it's a really boring movie instead of being a really boring video game. So the other side of this is to use a full field stimulator, a stimulator that encompasses the entire eye. You get to see the back, and then you see a flash of light. And by using flashes of different frequencies and timing them accordingly, we're actually able to pick up a component that corresponds to the ganglion cell function. So we did this in patients who have other diseases, such as glaucoma, where the nerve itself is completely destroyed in one eye versus the other, and you can see that there's a significant difference in the component that they're picking up and the size of that response. This was also done in patients who have another disease called idiopathic intracranial hypertension, and it was shown to correlate with those visual fields I know you all love so much. So we did some testing in patients who come out to Los Angeles, and again, we were able to detect a response. This response was significantly smaller than that from the control population when we went back and looked at normal controls. And this led us, through some help from Stealth Biotechnologies, to do a study down in Brazil, where we were able to look at patients in Brazil who are all from the same pedigree. They all carry the 11778 mutation. This has some advantages because when you're dealing with a group of individuals and you have a lot of different mutations, you have a lot of different environments and other things that impact it, they're going to present differently and they're going to have different features from the disease. So these patients have been followed annually since 2001. And when we were down there, we were able to get some recordings from them. And again, what we found was that we had a difference between the controls, the carriers, and the affecteds. And what was quite interesting to us was that some of the carriers also showed a much slower response than what we saw in the controls. Now, this is an area of quite a great deal of interest because we don't know what's going on with these individuals. We only have had the opportunity to record from them on a single occasion. But understanding this might give us a way to be able to either predict whether or not somebody's going to become affected or highlight the individuals that are more likely to become affected. And it also has implications for other mitochondrial diseases as well, because if we're showing that there's this range that's increased in individuals who carry any other types of mitochondrial diseases, it can be useful for outcome measures for other diseases also. We also found that if you cut off at -20, none of the normals had a response below -20, and none of the affecteds had a response above -20 microvolts. And this was reproducible across increasing light intensities. We've concentrated on the 1 candidate per second per meter squared, and if you ask any of the people who've been to LA, they'll tell you that this is bright enough, so certainly going up there was not necessary. And so this is where we're heading now. And what we're trying to do is we're trying to validate this. We're trying to understand this response more. We're trying to see in patients who are carriers as well, whether or not we're picking up the same variability, and we want to see across different mutations. The reason we're doing this is because we're trying to come out with good, concrete outcome measures so that when we do clinical trials and we do clinical studies, we don't miss a response because our testing technology is not good enough. So that's the purpose of this project, and it's going to be run out of 2 centers. I'm actually cross-affiliated in a way between the University of Ottawa Eye Institute and the Doheny Eye Institute at UCLA. And so both these sites will be recruiting patients, and we're going to be, our goal is to get 100 subjects from various mitochondrial diseases, especially Leber's. And certainly this project would not be possible without the support of LISA, the UMDF, and other people that you've met today. I just wanted to say, though, that as in terms for us, the most inspiring thing is you. So I would give yourselves a round of applause.